Hey class, here's a short video a little bit about the setup of the midterm lab. So you can see here, first of all, as you see, I actually printed out the lab manual. Now you may be very comfortable using electronic copies, but sometimes it's really handy, especially if you're in your kitchen and you don't want your device to get dirty or anything. So before we get started, remember that you are supposed to take pictures of your experiment before, during, and after, and have that card which has your name, your signature, and the date on it, so that way we know that it's you who did the experiment. Also the manual, it has all kinds of information, and I do want you to read the introduction and everything about cellular respiration. But the part that really interests us, if you wonder about the unit, it has to do with how we measure units in science. It's the metric system. And also there is a discussion about uncertainty, and what it means is that one experiment is not an experiment. We usually do replicates, and although in this experiment you're going to have only one replicate, but it's good to make multiple measurements. But anyway, this is the part where it describes how you set up your experiment, and you're going to need, for starters, bottles, and they can be glass or plastic. I don't like the plastic part, but it's simple and it's easy to get. And what is important is that they are all the same type. They all contained the same thing, and ideally water. And they are transparent, so you can take measurements from the outside. Then you need yeast, and any kind of dry yeast will work. You need sugar and you need water. One thing about experiments in general is that you want to have the everything set up ready before you start your experiment. Is a little bit like cooking a big meal. So for example, in this case, as you can see, I got the yeast here, I got the sugar here, and I got the water here. Now, if you wonder how many bags or packages you need, so this is what was in one packet of, let me just get the teaspoon of yeast, and you're going to need 1 teaspoon of each, and this is very hard to do with one hand, but obviously you need to fill it up. But seems like one of those packages is going to be enough for 2 samples. So you may want to have a few packages. And with sugar, you're going to need also 1 teaspoon for each sample. Now, I'm going to stop talking now, and I'm going to just set up the experiment. But again, you want to have everything ready to go, so then you just go and add the yeast to each of your bottles and add the sugar to each of your bottles, and then you add the water. And for the water, you need a 1/4 cup measure. Unfortunately, mine isn't a very nice one, but you want to be clear where is your measure. If you have one of those exact measures, that's good, but anything that can measure 1/4 cup would be fine. Okay, a couple more very important things. One, that you need to know which of your bottles is which, so you have to label them. And because many, 4 or 5 of bottles are going to go in a water bath, if you put tape on them, it may fall off. So if you have a Sharpie, probably the best way is just to write a number and put the number on the top, so it's not, it doesn't interfere in your measurements. So this is the could be number 1 or number 2 or number 3. So this has 2 teaspoonfuls of the yeast and 1 of sugar, and I will be adding water to it. And this one is the one which is number 4, which has 1/3 of teaspoon. Now the set I have actually doesn't have 4 1/3. It only has for a quarter or half. So to be honest, it is fine to use 1/4. You want to see what happens if you add less sugar. And last but not least, you are to set up 6 bottles, and then number 6 is up to you. So in that case, you can try different things. So you can probably, you will have the water. I would still add the yeast, but you can add either a different source of sugar or a different source of nutrient or something else. And or you can do the same with a different temperature. So in this case, we are using room temperature and a warm water bath. So you can, if you want, you can try a colder temperature. But again, it's up to you, or you can set up one exactly like the others, but then you can add, let's say, drops of vinegar or drops of ammonia and see how the pH changes. So it's really up to you. Okay, so the bottles are ready to go. In this case, again, I have only 2 bottles. In real life, you will have a minimum of 4 bottles or maybe 5. One recurring issue with the water bath is that as the water heats, the bottles tend to tumble. So you may want to put something around to hold them in place, or look for some kind of pot that it's snug so the bottles are not falling over. During the process. And another thing is that you need to time it. So ideally, you don't want to leave your bottles waiting for the water to be warm. So this water should be warm and ready to go by the time your bottles are ready. So as you can see, we are halfway through the experiment and the balloon is growing. I'm not going to wait till the end, but I just wanted to demo how you measure. You are supposed to measure the circumference of the balloon. So if you have a measuring tape like this, you can do it with the tape directly. Just go around and measure it. Remember to use the centimeter side. If you don't have a tape like this, you can use a string and then just, measure it in a different ruler. And then the other thing is the long axis. So this is a little bit harder, but try to always start with the same point and end in the same point. So the data recording, you are going to first describe the general conditions, what kind of yeast, what kind of temperature, etc. Then you have a number of tables in which you will describe what happens to the balloons, and you're going to be recording measurements. So one of the measurements is the height of the yeast solution before and after, and then the size of the balloon. And for the size of the balloon, you will be measuring, as I was showing you, the circumference and the long axis, and then you will calculate, according to a formula, the volume of the balloon. And these tables, these are not, you don't need to include this in your report because you are going to make graphs, and I will be talking about this during the Zoom meeting. But again, just be sure that you write all these parameters down, and also your observations. So the last thought here is that when you start the experiment, you may have a hypothesis, you're expecting certain results. And you may not get those results, and that's perfectly normal and it's perfectly fine, and this is the nature of science. So, we have a hypothesis, and sometimes it turns out that way, and sometimes it doesn't, and that's how we gain knowledge. So if you get results that they are unexpected, that is fine. You're going to write them down and you will be discussing them in your report. But just want to make you aware that, again, there is no such thing as a perfect experiment, especially if this is new for you, and especially if this is one experiment. So just record your observations, write down your data, and then in the report you can discuss why did you get those, what were your expectations, and what happened, and why do you think that happened. And again, this is just the nature of science.